I want you to just take a second and imagine a high-stakes clinical scenario. Picture a patient in the ICU, and they are really struggling to breathe.
Yeah, it's one of the most terrifying situations in critical care.
Right. And the real tragedy here isn't necessarily that their body is, failing to fight off a bacterial infection or something like extrapulmonary sepsis.
No, the actual danger is often the body's own immune system. It overreacts so fiercely that it ends up accidentally destroying the lungs in the process.
Exactly. And we're talking about acute respiratory distress syndrome, or ARDS. If you've been following medical news over the past few years, you've probably heard of it. It manifests as acute hypoxemia, which is just dangerously low oxygen levels in the blood, and bilateral pulmonary edema.
Which basically means both of your lungs are rapidly filling with fluid.
Right. And today, our mission for this deep dive is to look at exactly how our own immune cells execute this kind of friendly fire, how they destroy that really delicate air-blood barrier where oxygen actually enters the bloodstream.
It's just a mechanically devastating pathology. And the primary instigators of all this tissue destruction are neutrophils.
Which are the white blood cells, right? The body's first responder.
Exactly. They rush to the site of the infection.
Mm-hmm.
But the damage in ARDS doesn't come from them simply like engulfing bacteria like a Pac-Man?
No, it comes from their ultimate weapon, this sort of suicidal mechanism called neutrophil extracellular traps, or NETs.
Right. And we have this incredible source today. It's a groundbreaking paper recently published in Cell Death and Disease. And they mapped out this whole destructive process.
And it's wild because they didn't just map how the traps destroy the lung. They actually found a previously unknown molecular doorway, basically, that lets these tracks escape the cell in the first place.
Which is a huge deal for potential treatments. But before we get to the doorway, we should probably explain the weapon itself.
Yeah, let's unpack that. For everyone listening, if you try to visualize a microscopic Spider-Man web, you're pretty close.
Yeah, that's a great analogy. But this web isn't silk, right? It's literally constructed from the cell's own unspooled DNA filaments.
Wait, it uses its own DNA as a weapon?
It does. It's wild. And the web is heavily coated in these highly toxic acid-like proteins.
Okay, so what exactly is the acid on this web? Let's not skip any details here.
Sure. So to understand why it's so damaging, we have to look at the specific granular and cytoplasmic proteins coating that DNA mesh. First, you have citrullonated histone H3.
Or CYT-H3 for short.
Right. And CYT3 plays a massive role in unpacking the tightly coiled DNA in the first place, so the web can actually form. Then you have myeloperoxidase, or MPO, and neutrophil elastase, NE.
And normally these are good things, right?
Oh, absolutely. In a normal, localized infection, these are powerful enzymes designed to degrade bacterial cell walls. They are vital. And the structural backbone holding all these toxins together is cell-free, double-stranded DNA, or dsDNA.
So the cell essentially weaponizes its own genetic code. It vomits out its own toxic DNA to trap invaders.
Basically, yeah. But there is a strict mechanical timeline to this. It's a process called NETosis. A cell doesn't just, spontaneously erupt.
Right. It's not just a random explosion.
Exactly. It's a highly orchestrated, stepwise progression of a nolytic cell death. The cell literally dismantles itself systematically. First, it undergoes cytoskeletal reorganization.
Which, if you think about it, makes total mechanical sense. If the cell is getting ready to eject a massive internal web, it has to break down its internal actin scaffolding just to make physical room.
Yeah, it's a mechanical necessity. So once that scaffolding is cleared out, you get chromatin decondensation. The tightly packed DNA inside the nucleus loosens up and expands.
Like unspooling a giant ball of yarn.
Exactly. And following that, the nuclear envelope itself disintegrates. This is really the point of no return.
Because that's where the DNA physically mixes with the toxic enzymes.
Yes, the DNA mixes with the MPO and the NE out in the cytoplasm. It all builds up pressure until the final lethal step, plasma membrane rupture, or PMR. The cell membrane bursts, and this huge toxic web is deployed.
Okay, but wait. This brings up a really fascinating biological paradox for us to think about. If NETs evolved over millions of years to trap and kill pathogens, why are they suddenly the bad guys in a disease like ARDS?
It's a great question.
Shouldn't a patient with a severe lung infection actually want more of these webs to catch the bacteria?
In a localized, highly controlled scenario, yes, absolutely. But in the exaggerated systemic inflammatory state of ARDS, these NETs act as massive damage-associated molecular patterns, or DAMPs.
So the body's own alarm system starts interpreting the weapons as the invaders.
Exactly. Because these webs are so large and so highly cytotoxic, when neutrophils release them in these huge, unregulated quantities, it triggers a catastrophic inflammatory cascade.
They set off the alarm bells.
Very loud alarm bells. They activate what's called the NLRP3 inflammasome within surrounding cells, which acts like a blaring biochemical siren, recruiting even more inflammation.
And that's what destroys the lungs.
Yes. Ultimately, these toxic webs physically degrade the alveolar epithelial cells, that fragile, one-cell-thick lining of your lungs. They obliterate the air-blood barrier, which directly causes the fluid buildup and oxygen starvation you see in the ICU.
Oh, wow. Okay, so before we move forward, let's just do a quick recap checkpoint of these first few minutes. We're dealing with ARDS, which is characterized by a destroyed air-blood barrier.
Right.
The culprits are neutrophils undergoing the suicidal process called NETosis to deploy toxic webs armed with enzymes like MPO and NE. And to actually deploy the weapon, the cell must undergo plasma membrane rupture.
That's the perfect summary.
So here's a profound therapeutic dilemma for you listening at home. If you were trying to design a drug to save that ICU patient, do you try to stop the neutrophil from manufacturing the weapon entirely, or do you just try to weld the armory door shut so the weapon can never be released?
In this paper, they focused entirely on welding the doors shut.
Which is brilliant. But to do that, they had to solve this glaring mechanical mystery that the scientific community had fundamentally misunderstood for years.
Yeah, they had to figure out how a massive, complex web of DNA actually gets out of a microscopic cell.
Right, because the conventional wisdom always felt, I don't know, a bit mechanically flimsy. The old model argued that plasma membrane rupture was basically just a passive osmotic event.
Yeah, the prevailing theory was that pore-forming proteins, specifically one called Gasdermin D, or GSDMD, and another one called MLKL, they poked tiny little holes in the cell membrane.
Like poking a hole in a water balloon.
Exactly. The assumption was that these tiny holes disrupted the cell's osmotic balance, water rushed in, and the cell just swelled up and popped passively.
But if we actually look at the physical dimensions here, the math does not work. The paper points out that Gasdermin D pores have an inner diameter of roughly 18 nanometers.
Right.
Meanwhile, these nets are assembling into these massive structures. They form clusters of 20 to 50 nanometers, and the entire meshwork can expand up to 200 nanometers wide. You cannot squeeze a 200 nanometer structure through an 18 nanometer hole.
No, it's impossible.
It's like trying to parallel park a commercial school bus in a bicycle lane. The physical clearance just isn't there.
I love that analogy. And beyond the theoretical math, the pharmacological evidence didn't support the old model either.
What do you mean?
Well, when researchers applied chemical inhibitors in the lab, like blocking MLKL with a drug called GSK872, or blocking Gasdermin D with disulfiram, the neutrophils still released massive amounts of extracellular DNA.
Wait, really? Even with the pathways totally blocked?
Yep, the chemical locks were in place, but the webs were still getting out. It proved there had to be an unidentified executioner protein out there actively tearing down the cell membrane.
But hold on, if scientists were so convinced that MLKL and Gasdermin D were the culprits for all those years, how did everyone miss the real executioner? Was it just like hiding in plain sight?
It was hiding in the sheer complexity of the immune response. To find it, the researchers had to use incredibly advanced tech. Specifically, single-cell RNA sequencing and hotspot analysis.
Break that down for us.
So instead of just guessing and checking individual genes one by one, they analyzed the algorithms of gene expression across thousands of individual cells all at the same time.
Wow, like a massive genetic dragnet.
Exactly. They were looking for a network of genes that activated in perfect lockstep with the biological pathway of NETosis during a lung injury.
And they found one.
They did. They found a specific gene network, which they called Module 8. And sitting right at the center of Module 8, highly expressed almost exclusively in pro-inflammatory neutrophils, was a protein called ninjurin-1, or NINJ1 for short.
NINJ1. And the crazy thing is, NINJ1 doesn't just passively poke a tiny hole to let water in. It operates on a completely different mechanical paradigm.
Yeah, it's totally different.
The researchers describe it as an active executioner using a cookie cutter mechanism, which I think is just a brilliant way to visualize it.
It really is. Unlike Gasdermin D, which creates that fixed, tiny puncture, NINJ1 proteins actively group together in the membrane. They physically carve out entire large-scale disks of the cell wall.
So they just jettison these massive chunks of the membrane.
Yes, creating these gaping, irregular holes that easily provide enough clearance for the massive 200-nanometer nets to escape.
It's almost like a microscopic game of Red Rover. Individually, these proteins are just floating around, but when the cell reaches the climax of NETosis, they rush together, link arms, and physically tear a massive hole in the wall.
That's exactly how it works.
But, as someone reading this, I have to ask, how do we know this isn't just some phenomenon isolated to computer algorithms or petri dishes?
That is where the clinical translation comes in, and it's what makes this caper so robust. They actually tested peripheral blood from human ARDS patients in the ICU.
Real patients?
Yes. Specifically, patients who met the strict clinical criteria of the Berlin definition.
Which, just for context, requires acute onset, bilateral opacities on a chest X-ray, and respiratory failure that isn't fully explained by heart failure.
Right, the real deal ARDS. When they isolated the neutrophils from these patients and analyzed them, they found massive, statistically significant spikes in NINJ1 expression.
Oh, wow.
And furthermore, this NINJ1 was perfectly co-localized with those toxic web proteins we mentioned earlier, MPO and CYTH3. The data from human patients struggling to breathe mirrored the single-cell RNA sequencing perfectly.
Okay, that is incredible. Let's do another checkpoint recap right here.
Oh, that's good.
So, bullet 1, we debunked the old assumption that an 18-nanometer gastromin pore could passively release a 200-nanometer web.
Right.
Bullet 2, single-cell RNA sequencing pinpointed NINJ1 as the central executioner of NETosis. And bullet 3, NINJ1 acts as a molecular cookie cutter, carving away huge chunks of the membrane.
Spot on.
So for you listening, think about this. Imagine your cells have different emergency exits based on the size of the threat. Does it make sense that nature built a highly specialized protein whose sole dedicated function is to blow the entire wall of the cell down?
It's an incredibly potent evolutionary tool for neutralizing localized threats. But as we see in ARDS, it becomes catastrophically bad when it's triggered systemically across the whole body.
Yeah, that friendly fire. But, correlation in human blood is compelling, sure. But correlation isn't causation. As a skeptical reader, I want undeniable proof. If we physically remove this specific cookie-cutter mechanism, does the lung actually survive the inflammatory storm?
Well, to prove that, you have to move from human observation to a highly controlled genetic environment. So the researchers engineered neutrophil-specific conditional knockout mice.
Okay, let's clearly define that genetic technique for everyone. They didn't just breed a mouse that completely lacked NINJ1 everywhere in its whole body, right?
No, absolutely not. A systemic knockout like that could cause massive, unforeseen developmental issues that would just muddy the data.
Right.
Instead, they utilize what's called the ILI6G Cre recombinase system.
And how does that work?
So they crossed floxed NINJ1 mice, where the NINJ1 gene is flanked by specific genetic markers with ILI6G Cre mice. The key here is that the ILI6G promoter is only active in neutrophils.
Ah, okay, so the Cre enzyme acts like a pair of genetic scissors, but it's only cutting out the NINJ1 gene in the neutrophils.
Exactly. Every other cell type in the mouse's body retains normal NINJ1 function. Only the frontline immune soldiers were disarmed of this specific exit mechanism.
Man, that is surgical genetic precision. So they have this pristine mouse model. They take the knockout mice alongside regular wild-type control mice, and they induce acute lung injury by administering LPS.
Right, lipopolysaccharide. It's a highly toxic component of bacterial cell walls known to trigger massive immune responses.
So what happens? What's the physiological fallout?
The wild-type control mice predictably developed severe ARDS-like symptoms and had really high mortality rates. But the conditional knockout mice, the ones missing NINJ1 only in their neutrophils, showed a profound survival advantage.
Really? Just from removing that one door?
Yes. Clinically, they maintained normal posture, activity levels, and they even had glossier fur. But the most striking evidence came from the internal imaging and the blood work.
Oh, right. The micro-CT scans. The scans of the wild-type mice showed dense, fluid-filled lungs.
But the micro-CT scans of the knockout mice showed beautifully clear, well-aerated alveolar spaces. The structural integrity of the lung was totally preserved.
That is mind-blowing.
And systemically, their blood work revealed drastically attenuated inflammatory markers. We're talking massively reduced levels of tumor necrosis factor alpha, interleukin-1 beta, and most importantly, a sheer drop in circulating MPO dsDNA complexes.
Meaning the toxic webs simply weren't making it into the bloodstream or the lung tissue.
Exactly.
Which brings us to my absolute favorite part of the paper, The scanning electron microscopy or the SEM images?
Oh, they are visually stunning.
Because this is where the physical reality of the mechanism snaps into focus. Think about it. If you have a neutrophil that has undergone all the internal steps of NETosis, it's dismantled its scaffolding, unspooled its DNA, mixed it with the acid, but it physically lacks the NINJ1 protein to cut the exit hole. What happens to all that internal pressure?
The SEM imaging captures this phenomenal cellular state known as ballooning morphology.
Ballooning.
Yeah. The cell membrane stretches, distends, and actually bubbles under the immense internal pressure of the expanding DNA web. But without the NINJ1 cookie cutter to sever the membrane's structural integrity, it absolutely refuses to pop. The plasma membrane remains totally intact.
So the entire toxic web is fully manufactured, heavily armed, ready to cause ARDS, but it's effectively trapped inside a microscopic bubble.
Yes, the surrounding delicate lung tissue is completely spared simply because the exit door was physically jammed.
Wow. Okay, time for our third checkpoint recap.
Go for it.
Bullet 1, we have the knockout mice proving survival. Bullet 2, drastic reduction in lung inflammation on the CT scans. And bullet 3, those incredible ballooning cells that physically trap the nets.
Yep, that covers it.
So for you listening, think about that imagery, the toxic web trapped inside a bubble. What implications does this have for treating autoimmune diseases? It's a huge question.
The therapeutic potential is massive. But identifying the target protein is only half the battle.
Right.
To actually design a small molecule drug that you can safely give to a human in the ICU, we have to zoom all the way down from the cellular level to the atomic architecture. We need to understand exactly how those individual NINJ1 proteins link arms to form that cookie cutter ring.
Which involves a biochemical process called oligomerization. Let's not skip over this. How does it work?
Sure. Oligomerization is the process where single protein units, known as monomers, bind together to form a much larger functional multi-protein complex, the oligomer.
Okay, so to prove this was happening during NETosis, the researchers used two different types of protein gel electrophoresis, right? Native page and SDS page.
Exactly.
So why do we need both tests? Does native page give us a look at the machinery while it's fully assembled and running?
Yes. Native page is non-denaturing. It's a gentle analysis that keeps the functional protein clusters totally intact. When researchers ran activated neutrophils through this test, they didn't find single NINJ1 proteins. They found massive, heavy protein rings weighing between 180 and 300 kilodaltons.
And the SDS page test is the opposite. It's a harsh chemical test that acts like a mechanic, tearing down a car engine, breaking all those non-covalent bonds so you just see the individual parts lying on the garage floor.
Precisely. SDS page completely denatures the proteins, and when they ran the samples through this test, those massive 300 kilodalton rings vanished, and only the small individual NINJ1 monomers remained.
Proving that they assemble into the ring.
Right. Comparing the two tests confirmed that during the climax of NETosis, these monomers rapidly self-assemble into massive oligomeric rings to execute the membrane rupture.
But they didn't stop there. They wanted to find the exact molecular hinges that allow these monomers to snap together.
This is where it gets really granular. They used a technique called site-directed mutagenesis on a human neutrophil-like cell line, DHL60, to alter the protein's genetic code literally one amino acid at a time.
Talk about finding a needle in a haystack.
Yeah, but they did it. They systematically narrowed it down to two highly conserved amino acids on the NINJ1 structure, lysine 45, or K45, and asparagine 60, N60.
Okay, so going back to our Red Rover analogy earlier, if the NINJ1 proteins are the players linking arms to break the membrane, K45 and N60 are the literal hands grabbing onto each other.
That's a highly accurate way to frame it, yeah. The researchers purposefully mutated those specific attachment points. They changed lysine 45 to a glutamine, creating what they called the K45Q mutant. And they changed asparagine 60 to an alanine, creating the N60A mutant.
So they basically chemically amputated the hands.
They neutralized the binding affinity, and the results were absolute. When they triggered NETosis in these mutant cells, the native page tests showed zero oligomerization. No massive rings formed.
At all.
None. The cells defaulted straight into that protective ballooning state, and the release of extracellular DNA plummeted. Without K45 and N60, the cookie cutter just cannot be assembled.
That is fascinating. But wait, that raises a massive mechanical issue regarding the old theory. If NINJ1 is doing all the heavy lifting to actually break the membrane, what was Gazerman D doing this whole time? Was it just like a bystander?
Not a bystander, no. It's more of an early facilitator. The paper presents a unified temporal two-step model.
Okay, walk us through the two-step model.
So gastromin D activates early in the process. It forms its tiny 18-nanometer pores, which, as we know, aren't large enough to release a net. But they are large enough to allow small molecules like ATP and potassium to leak out of the cell. This leakage drastically alters the osmotic tension and the electrochemical gradient of the plasma membrane, and NINJ1 acts as a sensor for this stress. When the membrane tension shifts, the NINJ1 monomers are triggered to rush together, interlock via the K45 and N60 hinges, and execute the final catastrophic membrane rupture.
It's an incredible cascade. Gastromin D picks the lock, the NINJ1 is the battering ram that takes the door off its hinges.
Exactly.
Okay, final recap checkpoint. We've got the oligomerization forming the rings, we've got the K45 and N60 amino acids acting as the crucial hinges, and we have the temporal two-step model where gastromin D sets the stage for NINJ1.
Perfect.
For you listening, we've scaled all the way from a patient drowning in their own pulmonary fluid, down through the genetic algorithms, and finally landed on 2 single amino acids acting as a hinge. Which level of this biology fascinates you the most? It is just incredible.
It really is a profound demonstration of biological scale, and honestly, it fundamentally alters the therapeutic landscape for critical care.
Let's bring it back home to the clinic. Why does this deep dive matter for the future of medicine?
Well, as it stands today, a diagnosis of ARDS carries an unacceptably high mortality rate. Our interventions right now are mostly just supportive, relying on lung protective mechanical ventilation and, fluid management. We have lacked precise drug interventions because the immune system is so blunt.
Right. Historically, if you wanted to stop the neutrophils from destroying the lung tissue, you had to use broad-spectrum corticosteroids or heavy immunosuppressants.
Which is terrible if the patient is fighting an infection.
Exactly. In an ICU patient fighting off massive bacterial sepsis, suppressing their entire immune system is incredibly dangerous. You're leaving the gates wide open for the original infection to kill them.
And that is the exact paradigm this research shatters. By definitively proving that NINJ1 is the terminal physical mediator of net release, it gives us a highly specific surgical therapeutic target.
So we don't have to nuke the whole immune system?
Exactly. If we can develop a drug that targets NINJ1, maybe a small molecule that competitively binds to that K45 or N60 hinge, the neutrophils can still migrate to the lung. They can still phagocytose bacteria. They can even undergo the early stages of NETosis to neutralize intracellular threats.
We just prevent the final deployment of the toxic web into the extracellular space.
We just jam the cookie cutter.
That is phenomenal. I mean, imagine in the very near future, standing in that same high-stakes ICU we talked about at the beginning. The monitors are chiming, the patient's oxygen is dropping, but instead of just waiting and hoping the ventilator is enough, the attending physician administers a targeted NINJ1 inhibitor.
Down at the microscopic level, that drug acts as a molecular lock on the lysine 45 hinge.
The neutrophils still swallow the bacteria, they still build their toxic Spider-Man webs, but when the pressure peaks, the cells just balloon.
The lethal inflammatory storm is safely contained within billions of microscopic bubbles.
The air-blood barrier remains intact, and the patient's lungs are saved.
It represents a total paradigm shift in critical care medicine. We're moving from just managing symptoms to actively engineering the physical mechanics of cell death to preserve organ function.
It puts the power back in the hands of the clinicians. So as you go about your day, I really want you to hold on to that scanning electron image of the ballooning cell. All that highly toxic, destructive potential safely quarantined behind an intact membrane.
It's a powerful image.
And we focused on the lungs today, but runaway inflammation drives immense human suffering across the entire medical spectrum. Think about conditions like inflammatory bowel disease, neuroinflammation in Alzheimer's, or severe rheumatoid arthritis.
NETosis plays a role in all of them.
Exactly. So here is my provocative final thought for you. How many of those devastating pathologies might just be waiting for their own molecular cookie cutter to be discovered, mapped, and permanently jammed? It's something to think about. Until next time, keep diving deep.